Thank you. Even though I've done the internships, I don't consider myself such a technical person. Compared to other people who have even dove even deeper into technical stuff. I think
That can come later, though. I think the most important part is like interests, which you clearly have.
Yeah, thank you. Actually, I think we are a little bit similar in that we are at the intersection of art and science. At least communications and science, and sometimes that's a little bit hard for me because how do you define yourself, right? It's not like many people talk about artists who are also scientists or the other way around.
Yeah. Yeah, it's true. It's kind of hard to do 50-50, like, for real, because as a scientist, typically you don't have formal training in art, and they're like, oh, you can't really call yourself an artist. But if you're an artist and you're saying you're doing science, usually it's just doped like pseudoscience. So, and then to say you're doing both, you're not taken seriously in either field. So I'm trying to change that. Like, that's why I'm trying to do science and art, where it's just like intertwined. Like, you can't tell me that it's not art, and you can't tell me it's not science, because it's just both. It's a beautiful flower.
Welcome to the Biopunk kingdom. My name is Sofia Sanchez, and in this episode, I have had the enormous pleasure of chatting with Nick Desnoyer, a plant scientist by day at the University of Zurich and a plant artist by night. If you're on science Twitter, you may recognize Nick as the guy who genetically engineered Arabidopsis flowers to go from white and relatively simple to pink flowers that had never existed before in nature. He has also more recently engineered Arabidopsis leaves to have striking patterns that mimic those of other plants, sort of pink and green. Really cool. Go check those out in the links. All of these projects are part of Nick's mission to communicate the beauty of plant science to the public. In fact, he believes that plants are the best organism to teach people about biology. And here we really get a chance to talk about art and science. Where do we draw the line between those? Can we engineer other traits into plants, like even movement? Can we use epigenetic tools to reprogram plants in other ways? What would be a future plant biotech tool that Nick would be blown away by, say, if he traveled 50 years into the future? What are his next steps for these educational projects? What are some other even crazier ideas to engineer plants? How is biology different as a medium of expression compared to others? And whether we can build something like a ChatGPT for flower design, a tool or platform or interface through which people just even draw or describe the flower that they would like to create, something that had never existed before in nature, and we can get the genetic or biological blueprint to actually grow it in real life. Nick is one of very, very few flower genetic designers around the world. So this conversation is very unique. He has become a great inspiration for me, and I am very glad to share his story to inspire more people to do different things in biotech. So if you also want to have a weirder biopunk future, follow the Biopunk. Tell your friends about it. It's all free. I'll leave the links in the description below. And now, let's start. Hi, Nick. Thank you for being here. How are you?
I'm doing great. Very excited to be here. Thanks for the invite.
Sure. Well, a question that lets me know a person better is who their heroes are. And I would like to ask you about your heroes, whether they're into art, science, or the intersection of both.
Yeah. So actually, some of my heroes, it's kind of funny, are podcasters, kind of like yourself. So in the lab, you have a lot of mindless work that you do sometimes, like plating seeds or transplanting seedlings. And so I've listened to hundreds of hours of podcasts, mostly by Joe Rogan and Lex Friedman, who invite really interesting guests on. who kind of tell their stories. Um, and they're just really great podcasters and just have great philosophy on life. So those are kind of my heroes, but I would actually say most of my heroes are fictional. So I look up a lot to like the heroes that I play in like video games as a kid. So like Sora from Kingdom Hearts or Cloud Strife from Final Fantasy, or like actual superheroes like Spider-Man. Um, Actually, even as a kid, I wanted to be a superhero, and I like enrolled in Taekwondo and got like ninja weapons and stuff. And as a kid, I even made a like a life promise to myself one night that the feeling I felt of like wanting to be a superhero and all of that imagination I had, I don't want it to ever go away. And I've held that promise since I was like 10 years old.
What is it about these heroes? Maybe is there something common between Lex, Rogan,
Yeah.
and the superheroes? Yeah, that you.
Yeah. Yeah. I would say that they're unshakable, that they like in the face of defeat, they just smile. So like Ash Ketchum, he's actually, I would say Professor Oak is maybe like my ultimate hero. I want to become like a Pokemon master, but kind of like Pokemon researcher, right? And Ash, he's just never shaken and always optimistic. And I think that's true for all those heroes that I mentioned.
What about your hero plant or flower? Because you've, of course, worked a lot with Arabidopsis. Are there any other plants that, because of their aesthetics or maybe other intrinsic qualities, you admire, either even in a philosophical sense or in whatever way?
hmm Yeah, it's funny you say hero plant. So the hero plant for me would be Arabidopsis thaliana. I think it gets bullied a lot in plant science because people think it's boring or that it's just like small and doesn't really compare to like real crops that we eat. Um, but there's so much knowledge about it, like 50 years of dedicated research on this single plant. And there's so much you can do with that knowledge. And none of it has really been applied because there's no real use for Arabidopsis as a crop. Like no one's going to eat it. But if you kind of put those together for an art project or an educational project, then it can really transform into kind of a medium for art. And of course, I'm inspired by a lot of plants in nature here. In Zurich, we work in a beautiful botanical garden. So actually, you're kind of forced to walk through a beautiful garden every day before you go into work. Um, there's one that really inspired me. It's called Saxifraga rotundifolia. It's a native alpine plant here, but it produces pretty small flowers, like ones you wouldn't even notice. But if you look up close, it has spots that extend down the petal. They're like perfectly spaced. And as you go from the base of the petal to the tip of the petal, they transition from like purple to yellow. So it's got like all the properties that you would want in kind of like a designer flower. It's got spots and it's a beautiful pattern and it has all the colors.
Very nice. I suppose you can get very focused on things that people wouldn't normally focus on when you're a plant scientist, right? You start to look at the world differently. But in your case as well,
Yeah.
like you already had some of these interests since you were a kid. So have you noticed those deepen, or how is your relationship with nature in that sense?
Yeah, totally. It's completely changed in like the past year, I would say. As a kid, I was never super interested in plants. In fact, I'm kind of embarrassed that I don't know the names of a ton of plants, kind of like my mom does, or when my mom asks like, what's wrong with my plant? Does it need more water or something? I don't really know. But since I got kind of into designing the flower, now I look at plants completely differently. I see them as inspiration and think of them as evolutionary innovations that I want to try and kind of recapitulate in the flower that I'm working on. So, like for example, with Saxifraga, it has these beautiful spots with different colors. It's like, how can I make that in Arabidopsis? And I think just that kind of thought experiment is really powerful. And that's why I want a major objective of this project to be kind of sparking imagination in students. So as I mentioned, Arabidopsis probably doesn't have any application as a crop, but I think the design of its flower kind of utilizing all that knowledge we have and just kind of thinking about how we can recapitulate those beautiful patterns we see in nature, that can be a powerful tool for getting students interested in kind of working with the knowledge that they're learning.
I love that. And it's interesting that you sometimes refer to this project and plants as a new medium of expression. Was the Arabidopsis project the first time that you thought of biology as a medium of expression and not really? Because I know you've done some very cool images in the past as well, and this is more like photography and videos, illustrations as well. But yeah, let me know a little bit more about your thoughts about art and biology.
Yeah, another excellent question. So no, it's not the first time I thought of plants as being the medium for the art. Because before that, I was really into time-lapse photography of plants.
Yeah.
And what I realized is when you stick a camera on a plant and start to film it over time and then compile it into a time lapse, they start moving in ways that you never knew they did, right? You're used to just kind of looking at static time points. Like I had no Arabidopsis does this crazy circummutation, sometimes multiple times a day. It's super wiggly guy. Other guys just stand like very stationary. But another interesting aspect of that is their movement, their behavior over time kind of reflects like their health and like how they're quote unquote feeling in the chamber. For example, a thing I quickly notice is that if you train Arabidopsis on a light cycle of 16 hours daylight, 8 hours night, and then you move it into a chamber with 24-hour light, so it's essentially getting no sleep, it almost goes completely infertile. And the flowers stop opening, it stops getting this circadian rhythm. And you can kind of like feel the emotion of the plant through time lapse. So that really inspired me. But in that case, the medium isn't really the plant, it's just the subject. But when you start to genetically modify the plant, then the plant actually itself becomes the medium. And I think this is really going to be kind of the next revolution in flower art. And flower art really goes back very far. I mean, for decades, it's been the subject of a lot of art with different mediums like painting. You're probably familiar with paintings by Frida Kahlo or Monet or Georgia O'Keeffe. They all kind of became obsessed with the flower, and we can maybe get into why that is. But there's also more modern artists like Takashi Murakami, who uses digital media of the flower. He's got this kind of super flat style. He designed album covers for Kanye West, and he even kind of has like designer flowers. But I think using the flower itself, shaping it, that is going to be kind of the next movement in flower art.
You touched a little bit on your goal and mission with the Open Flower Project. So, knowing that this can be both a tool for teaching students or even the quote unquote general public about biology, but it can also be a medium of expression. Tell me more
Totally.
about how you envision this project to develop.
Yeah. So as a medium for expression, it's quite difficult because you're still pretty limited in what you can do. So it's at its very early stages. Like you can envision the kind of flower that you want to make. Maybe you can even prompt ChatGPT to come up with an idea of what you want to make. But we're very far from that. Like if you want to choose the number of petals of a flower has, you can't really do that. You can make more by mutating certain genes or by doing kind of horizontal gene transfer, taking genes from other plants and putting them into Arabidopsis, kind of like we've done with Ruby. But the palette is really limited. I listened to a podcast with Sebastian Kotsioba, who I'm sure will also talk about big inspiration for me. Another flower designer, but been doing it for much longer, for like 12 years in his home lab. And he equated it to kind of having a painter's palette. And all we really have right now is pink. And
Wow.
for me, pink does not speak to me. In fact, like I'm kind of embarrassed that all the flowers I make are a bit girly. Like I want to work with black. I want to make like a gothic flower. That is the kind of flower that I feel I could express myself more with. but we don't have those colors yet. But I think they'll come online relatively soon. And I got some ideas.
Yeah, actually, I want to dive a little deeper into the ChatGPT question.
Yeah.
Would it be possible in any way right now to create some sort of UI, digital page or app where
Hmm.
people can, click a button and say, I want, to knock down this gene or I want to add this color. And I know the palette is very limited right now, but could we have something like that, or could we even, yeah, what are some tools available that can maybe make
Yeah.
this flower design even more, even easier for people to use?
Yeah, another great question. So all of the things we have for flower design are hidden deep within papers, right? You say, oh, I want to make the petal a certain shape. Now you have to dig through the literature of Arabidopsis, which is tens of thousands of papers. But now with LLMs and things like ChatGPT coming online that can maybe extract data from these papers, it can maybe pinpoint you to the papers you really need to read in order to make those uh, phenotypes. As for having ChatGPT kind of come up with an idea for a new flower, I have explored this a bit, and it's very good at coming up with pictures of flowers that don't exist, probably because the internet is just saturated with pictures of flowers. I don't know this for sure, but I imagine like people take probably mostly pictures of cats and dogs, and then second to that is flowers or flipped. So ChatGPT is really good on the data that it's been trained on, and there's a ton of flowers. In fact, there's even a Twitter channel. It's called The Flower Show, and sometimes they do like a little quiz where they show four flowers and they say which one's actually real, and
Wow.
it's hard to tell.
I've encountered some of those, let's say, generative AI flowers, and I got really disappointed when I actually looked in more detail at the image and said like, "Oh shit, this is not real." It was it was sad, but I do hope that in the future we can probably bring those to life.
Yeah, it really likes to add water droplets to petals for some reason. So
Yeah, I've seen it.
removing those is difficult. What I've been playing around with is making vectors of flowers, so like line art in Illustrator. And with the Adobe package and the newest version of Illustrator, you can select that vector and do something called generative fill, where it kind of gets the idea of what you're trying to draw, and then it fills it in, which you can adjust with a prompt. Um, so I would like that to be kind of a tool as the package I'm trying to make with this flower design kit, that the students can also kind of draw or make up what they would like to
Mm,
make, and then they can play around with this generative fill.
very cool.
But then making it in reality, of course, is next to impossible. I've thought a lot about this because in terms of all the research on Arabidopsis thaliana, probably the most robust model is the ABC model of floral development. It's probably the best model we have for it. And I would have to guess that it's the subject with the most papers. And the evidence is also the most robust because it's kind of just black and white. It's like you mutate this gene, here's the phenotype, and they have pictures of the flowers in those papers. So it's not something like a gel or RNA-seq and whatnot. However, these pictures are mostly from like the 90s, so they're not super nice. So I want to get a lot of these genotypes, take really nice quality images of them, maybe even 3D models of them, and kind of feed that into an LLM maybe in the future to kind of train it to understand what certain phenotypes look like. It can actually predict what the phenotype of double mutants would be or triple mutants. It would be really cool.
Yeah, for sure. Something else that I wonder is apart from petals and maybe color, what are some other characteristics, features that you could engineer into a plant? One of the ones I'm excited about is movement, like plants like Mimosa pudica or others. I think it's called the telegraph plant, the one that actually moves in a way that is visible to the human eye. I would love to have something like interactive plants. You touch it and it reacts to
Yes,
your touch.
yes, 100%. Yeah, so I just kind of had thought experiment with a friend like a few months back, and we were just jotting down like the best ideas for designer plants. I'm like, what's the best one? Like something that happens instantaneously. Like you touch the plant and it like
Mhm.
moves away. But this I think would be extremely difficult for it to move that quickly, like kind of like Venus flytrap. The anatomy it
Yeah.
just is much different. But plants like Venus flytrap are just so fascinating. It's almost as fast as some of the slower neurons in animals, which is quite
Crazy.
mind blowing. So I think movement is very far off.
Mm.
But some more realistic things that actually make the plants interactive would be to use things like induction systems. So these are systems where you can add an inducer molecule. This can be a hormone, it can be ethanol, or it can be kind of an environmental stimulus like optogenetics. You can use light or heat to activate the production of some certain protein. And I want to kind of multiplex these induction systems so that you can add different stimuli to to change the plant in some way. Another thing I'm really interested in, which is now actually working in my hands, is induced variegation. So there's a really fascinating mutant called mutants um, that essentially with highlight, the plant induces kind of white sectors because all the chloroplasts die. But at some point in the white sectors, some of the chloroplasts, they revert back to wild type, and it's still not totally clear what this compensatory mechanism is, but you get these kind of stripes of green and spots of green. And then now that you have a white leaf, now it looks like a canvas. It's like a multicellular canvas. And so if you could add an inducer molecule, which I've done now, to like the epidermis of this white multicellular canvas, now you can actually use the leaf as living tissue. That's your your living canvas.
Is there a way to actually, let's say, draw the
Hmm.
variegation, meaning
Hmm.
to decide which of these chloroplasts die in a way?
Mm. That's another thing I've thought about a lot. So that was kind of the idea behind this dexamethasone inducible ruby that I made. So essentially, you add this hormone to wherever you want ruby, the production of this pink pigment to be. And I was a bit naive. I thought at first I could get like a needle and dip it in the dexamethasone and then draw like hearts onto the flower.
Mm,
But
nice.
because it's living tissue, it's much more complex. For example, the petals after they're fully developed, they're likely no longer transcriptionally active because if I have a mature petal and I add the dexamethasone, you don't get induction.
Right, and when they
Also,
are, they're
the
growing, so they
yeah,
mature.
so you got to get it earlier in the organ primordium, and also the dexamethasone diffuses away. But maybe if you use a different solvent, or you can make a conjugate to the inducer molecule that limits its diffusion, then you could draw on these. Just one last thing. Maybe you've seen white roses in a garden before that have pink spots.
Mm.
These look like they're infected, and they are. Well, to me, I first thought that they're like a freckled variety that breeders selected this so that they have spots. But then I learned actually later it's botrytis infection. So wherever the I think it's a fungus infects the petals, it makes this a localized anthocyanin production. And I think if you could figure out whatever effector molecule from the fungus is triggering this anthocyanin, then maybe with that you could go and draw onto the petals.
What about epigenetic modifications? One time, one of my plant bio mentors told me that
Hmm.
this was one of the areas that she was interested in and that probably people hadn't studied as much. I wonder if it would be tools
Yeah.
like, D-Cas9 or maybe some
Yep.
kind of elicitors that
Yep.
you would use to modify the plant's epigenetics.
Yeah, great question. So our lab actually studies epigenetics a lot.
Mhm.
I'm in the group of Uli Grosniklaus. He's studied epigenetics pretty much his entire career, and about half of the lab is dedicated to that. Epigenetics is really any change in gene expression that's not linked to a change in the gene code itself. So this can be like chromatin remodeling or DNA methylation. But if you take that really loose definition, it can also be these induction systems. So you add a molecule, and now the protein is being produced. So I think in a way this kind of is epigenetics. I hope that's not wrong, Uli. But I think with like epigenetic reprogramming, you have a bit more control over the gene expression, because right now we kind of just look at knockout mutants or wild type, and so you either completely ablate the gene function or you look at its full function. It's kind of just like black or white. But if you can finely tune its expression, then you can maybe get better phenotypes. Because a major problem is a lot of the mutants that I would like to have in the flowers, they're either embryonic lethal, so you can't even get a seedling, or they have like such defective vegetative growth that the flowers are already quite messed up because the plant just overall is sick. So if you could use epigenetics so that the mutation is really only manifested in the flower, that could be quite useful.
And are there any, let's say, specific or sort of visionary ideas that you would create with epigenetic modifications in flowers that wouldn't be possible otherwise?
I would say, yeah, using these induction systems. So for example, right now I look mostly at a mutant called agamus. So when you remove agamus or break the gene, you can no longer specify the carpels and the stamens, and in place you get sepals and petals. You can also stack this with another mutant called Superman, which is important for the boundary between the stamens and the carpels and floral termination. And so you get like then 200 petals. It's what I posted on Twitter recently. And I find this super fascinating because Arabidopsis has about 25,000 genes. We've looked at mutants of most of these genes, and most of the phenotypes, the plant just looks a bit sick. However, there's one that really stands out, which is pops up in forward genetic screens. It's like 1 in 10,000 mutants or something. And so there's like this really special mutation. It means a lot to me. I even got it tattooed on my wrist for many reasons that we could go into. But I find it even more interesting that, all right, this gene's super special. It's 1 in 25,000. But now you look at double mutants, and now the combination of different mutations becomes like, I don't know, like 1 million. And the agamus Superman double mutant, this one's spectacular. I mean, it looks like a zinnia or a dahlia, which I just find super fascinating. In terms of, yeah, using epigenetics to regulate their expression, I think this would be really useful for restoring their fertility. So these flowers are completely sterile. And so that means they can't produce any seeds, which is great for providing them to classrooms because they're quite safe, and maybe we can get to that later. The problem for me is that I have to propagate them as heterozygotes. And in fact, I'm using original alleles from an EMS screen that I can't even genotype, or at least I haven't. So I genotype completely by eye. So what I'm trying to do now is create an induction system where I can rescue their function. I can collect the seeds from these, and then all of these seeds are homozygotes for both mutations. And so I can distribute them. They'll all show the phenotype, but I can actually still restore their fertility and collect the seeds when I want. And this will get quite cool when you have different varieties, and then you can start to cross them and do real breeding, because even though Arabidopsis is quite inbred, at least the accession we use, we have quite a few other ecotypes that have different characteristics that I would like to integress.
If you could travel in time into the future, or if you could just, have a magic wand and say, I think this is the future tool that plant designers or biologists need, and that would change flower design forever, what would that tool be? It can be, imaginary. It can be something that you are already starting to see in the field, whether it's for imaging or genetic design, epigenetics. Yeah.
Yeah. So I find this question really interesting. Actually, it's one I always ask my friends, like, it's a bit different, but would you rather travel into the future or travel into the past? And if
Oh.
so, at what point would you want to go to? And I think that tells a lot about a person. Um, yeah, I find it interesting that a lot of people actually want to go to the past, but completely tangential. If I went to the future, what I would hope is that we have like plant growth chambers or time-lapse chambers that image the plants and can predict kind of their internal states. So using biosensors that can emit some kind of signal that tells you, oh, my plant needs water or it needs more sunlight. And I think this actually will come online relatively soon. There's a super cool company called Lightbio. You've
Mhm.
probably heard of them. They're selling this firefly petunia. This is a glow-in-the-dark petunia. It's auto bioluminescent. Has some genes from fungus that make it glow. But now I envision that this can be a whole new field of bioluminescence imaging in plants, as it's quite bright, at least relative to the luciferase that was been used before. And so I imagine you have like plant sentinels, perhaps in fields that they glow when they detect a certain pathogen or when they start experiencing drought. And then you have, yeah, like uh, cameras that can pick up this signal and kind of inform the farmers.
That's very cool and very interesting. I just thought about it in an art sense, and yeah, maybe since we already have a lot of different sensors for agriculture, there might be some. We might start to engineer sort of new interfaces between
Mhm.
plants as biosensors and then bringing that
Mhm.
to the digital world.
Yeah. Yeah. So my plants in a way are already kind of sensors. So I made this induction system where you add
Right.
dexamethasone and then they show Ruby. And so if you wanted to detect dexamethasone in the soil, there would be no reason for this, but you could use them already as a sentinel. So I imagine they could be engineered to be sentinels for more important compounds. And I had this vision that you have all these sterile agamous mutant flowers. They're all white, but they have a sensor for, let's say, heavy metals in the soil that
Yeah.
you want to find, or maybe even TNT. And you spread these out into a field. By the way, we're not doing this for anyone listening. Theoretically, you spread these out, and then you look for the pink flowers, and you know kind of what's in the soil, or have them be a readout, for example, of the microbiome because that's kind of the dark void, right? What's happening underground. And you could have the aerial organs kind of be a signal for what's going on in the underside.
This reminds me of one of the reasons flowers might actually be the best way
Hmm.
to teach people biology is that unlike bacteria, which I think have been more used in
Mm.
biotechnology education, they feel more familiar, right? When you think of bacteria, most people think of disease, and when they think of flowers, they think of the gift they gave to a loved one, right? So how would you think, if you have, actually, my question is directed to, have you already started teaching this course? If so, how have you seen people's opinions around genetic engineering or biotechnology change? Or if you haven't yet,
Hmm.
how do you expect those to change given the nature of the organism?
Right. So you mentioned this course. I'm not sure all the audience knows
Right.
what this is and what my intentions are there. So recently I wrote a grant proposal to make a kit to provide to universities to be used for practicals in teaching genetic engineering. So there's already hundreds of tools and kits available for things like teaching mechanical engineering and electrical engineering, but there's really nothing for genetic engineering. And I feel like we're really missing out there. But as you kind of highlighted, the problem is more with plants. There's already stuff that exists for bacteria. So I'm talking more gene editing in plants. Like for example, Sebastian, he made a really great collection of chromoproteins that are expressed in E. coli, and they make these beautiful colors, all the colors of the rainbow. And I found these to be really effective for teaching, and microbiology is great because it's very quick. And so I was trying to think like, how can we make teaching genetic editing in plants fast, right? Because if I want to change the flower, then I need to make my construct. I have to transform it into the plant. I have to recover the transformants, homozygous this, and it can be months of work. And so it's not realistic for students to do this. However, they, as I've mentioned several times, they can use induction systems, which act really quickly, where they can add an inducer molecule and get a phenotype within just 24 hours. And so I think the flower here is a really great basis for teaching this, in parts because these flowers are beautiful, and so they are something that already kind of attract people. As you mentioned, they're familiar with them. Often, if you start at the level of cells and molecules, in my experience with outreach, it just kind of goes over people's heads. But if you start with the flower and then transition into cells and molecules, then it's much easier. Another great thing about these is because they're sterile, they don't really apply to or appeal to this emotional aspect of kind of affecting the ecosystem, which is a major driver of why I think people are anti-GMO. So there's that, and then there's also the fact that they're not going to be eating these flowers, which is another emotional aspect that has led to the rejection of GMOs. For example, you may have heard of golden rice. This was a
Yeah.
humanitarian project where some really bright scientists they made the precursor for vitamin A, beta carotene, which makes a kind of an orangish color, and they put this into rice as a humanitarian project to provide to people in third world countries where vitamin A deficiency is a problem. But the marketing on this was not very great. And so for like 12 years, they could never get it out. In fact, I don't think they ever did it. There was a lot of campaigns against it. And so like a phoenix from the ashes, I want to make the golden rose. I want to take the same genes that they used and put it into my Arabidopsis roses and kind of use this as a platform for teaching like what went wrong from a much more safe and approachable way.
I've also been thinking about this concept of biopiracy in the sense of stealing. How could somebody steal bioart by
Mhm.
maybe cloning the plants through tissue culture or in any sort of way?
Mhm.
Because if we think about this as actually
Yeah,
developing as something that has a market, something that can be a business, design or flowers,
yeah, yeah.
have you thought about that? Is that possible at all?
Yeah, I've definitely thought about that because we started to talk about plants being the medium for art, but they're such a radically different medium. Like if you make a painting, no one can, I mean, someone can steal it, but there's just one original copy. But these are living organisms, they replicate. And so if you release one plant, someone can just propagate it and you can no longer make money off of that, which isn't actually the goal, by the way. But what the kind of upside of that is that they're kind of a legacy. So if you make a beautiful flower, now this can propagate longer than your lifetime. So even after you're gone, you have kind of this living legacy, this collection of flowers that you made. And that aspect I really like. Like some of humanity's greatest art, like the Great Pyramids, these were incredible monuments that in achievements of human civilization. And in fact, I think art is probably the best way of like standing the test of time. Like people don't really know what was happening day to day in Egypt, but it's the Great Pyramids, they kind of last and it's the thing that we remember. So often the thing we remember from the past is the art that humans made. It's not kind of what they were doing. But in terms of kind of monuments, these erode and degrade over time. Whereas if your achievement is a living organism, this can propagate for as long as you are a steward to it. Um, and
And
so
it'll also
that aspect,
evolve, and probably people will continue to modify it, right?
exactly. It's not like you would make a painting and then someone would take it and be like, I can make it better and paint over it. But with this, you can. So I can make a collection of these roses, and then someone can take it and improve it, and that's so cool.
Yeah, I imagine also in terms of intellectual property, we might have different
regulations or, laws coming into place, right? Because right now, it's not like you can own a whole organism, right? But you might patent some kind of modification. But then I believe as these modifications, as the palette gets broader and broader, I don't know. I think things will have to change.
Yeah. So actually, last Christmas, when I made the first post about the Arabidopsis rose, I was contacted by a few venture capitalists who were interested in turning it into a for-profit company. And I talked with them for like the majority of last year. I even got a position in New York, but I ended up declining it because I realized it gets much more into the legality. At some point, I would become more of a businessman, less of a scientist. And they just didn't leave a good taste on my tongue that it's all about the commercialization of this flower. It kind of steers away from the initial motivation. What I would much rather do is have a non-for-profit company where you can distribute these flowers and get flower design in the hands of other students. Um, I mentioned at the beginning of the podcast that I made a life promise to myself as a kid to remember how I felt as a kid, all the imaginative fantasies that I would build up, all that creativity. And I've always kept that, and the Rose project really re-sparked it, but kind of in a new way where I want to make a second life promise, which is to not just always keep that feeling close to my heart, but share that with others and try and get other people to feel the way I feel when I look at the flower.
That's very inspiring.
Thanks.
To close the episode, I'd like to ask you, let me see, I have a couple of questions. Is there something, first of all, is there something that I haven't asked you yet that you would like to share about your work, about yourself?
Yeah, like if you're listening and there's one thing that you take away from this podcast, if there's something that sets your heart on fire that you just completely love, go chase it and chase it without fear because you are so lucky to have that feeling. I think a lot of people can go their entire lives without finding that. And if you found that, you would be stupid not to follow it. You only got one shot. And let that kind of fuel your mission and don't steer away from the mission. Make a life promise to yourself to carry that out. And then you will become indestructible, just like the heroes I mentioned at the beginning of the podcast. And if you haven't found that and you feel like you're stuck in a hamster wheel, like just moving forward, but not actually getting anywhere, break out of that. Go do something that makes you feel uncomfortable and just start exploring and hope, yeah, that you find something like that. You might as well try.
Specifically for plant biology, is there anything you would say to not current plant biologists, but actually who will be the next generation of flower designers, maybe 50 years into the future?
Anything that I want to tell them. Embrace new ideas. I feel in science we can often be quite close-minded, especially the reviewers of grants. If you don't propose something quite conservative or safe that's for sure going to work, then it's much less likely to be approved. But I wish we embraced a bit more kind of crazy ideas, and you don't know where that's going to lead. So yeah, I would say keep an open mind.
Thank you so much for sharing about yourself, your work, and visions and advice for plant engineers and people who are just interested in this space, Nick. Thank you.
Yeah, thanks for having me. It was a great time. Yeah, nice meeting you, Sophia.
Bye, Nick.